All right, well, we're going to get started. Delighted to welcome all of you to our Grand Rounds today. Thank you for venturing out in this subtropical temperatures that we have in Madison. So I'm very excited about today's special guest speaker, Dr. Bruce Levy, who I've known for many years. Delighted he was able to make the trip out from Boston uneventfully and hopefully back there uneventfully. And to introduce Bruce, we're going to have Dr. Lauren Denlinger, our vice chair for clinical and translational research for the Department of Medicine and professor in the Division of Pulmonary and Critical Care.
Thank you, Lynn. It is my pleasure to introduce Dr. Bruce Levy. He is the Francis B. Parker Professor of Medicine at Harvard Medical School and a friend and colleague since at least 2011 as the chair for the NHLBI Severe Asthma Research Program. He has humble Midwest roots, grew up in St. Louis, went to med school and undergrad in Philadelphia at University of Penn and received AOA honors there. Went on to Brigham and Women's Hospital for his residency and fellowship, as well as a research fellowship in biochemistry at Harvard Medical School. He's had numerous clinical leadership roles over the course of his career, has been division chief for pulmonary critical care since 2013 and department chair for medicine at Brigham and Women's and physician in chief since 2022. His teaching includes mentoring well over 55 grad students, fellows, postdocs, residents, faculty who have gone on to academic careers, and he's the PI for the T32 at their institution in pulmonary. His research career is really illustrious, at least well over 150 publications in highly prestigious journals, Science, Nature, New England Journal of Medicine. He has 6 active grants, at least 10 patents, advisory boards for NIH and industry. He's the associate editor at New England Journal, as well as our top specialty journal, the American Journal of Respiratory and Critical Care Medicine, and has editorial roles in 3 basic science journals. He's had a career in terms of understanding the mechanisms for actively turning off inflammation. His title today is Unravelling Systems of Resolution of Inflammation in Asthma. Please help me welcome Dr. Bruce Levy.
Is that working? Great, thank you, and thanks for that. I wish my mother could have heard that. That would have been nice. It's really lovely to be here in what I would call a winter wonderland. You have more snow than we have in Boston, but it's very similar. And really to talk to you about asthma. This is one of the hubs for scientific thought about asthma with Bill Bussey here in the audience. It's so wonderful to see you, Bill, and Lauren and Nazar, many of your friends and colleagues here in Wisconsin. And so for me to be here and share information with you that how we're thinking about asthma, it's really a thrill. And I understand there's trainees in the audience, so thank you. I know you're probably very busy clinically, so I really appreciate you taking the time to join us. All right. For those of you who are signing in, here's the code. Some conflict of interest. As mentioned, I help to lead our severe asthma research program, sponsored by the NHLBI. And as part of that, we have multiple companies who are also helping to support the effort. And so that's what typically most of these are about. Nothing really directly relevant to the talk today, per se. Okay, that's the Brigham on a nice, clear, sunny day. Today's Valentine's Day, and we're talking about asthma. So Saul Permit is known to have defined asthma in this way. Asthma is like love. Everyone knows what it is, but no one can agree on the definition. Well, that thought advanced a little bit. I like this definition. It's simple and straightforward. Asthma is a chronic inflammatory disease of the bronchial tubes in which the airways constrict too much and too easily to a variety of environmental stimuli. I thought that was straightforward, but of course, experts got together at the NIH and put together the expert panel, 3, and I have to take a big deep breath for this one. Asthma is a chronic inflammatory disorder of the airways in which many cells and cellular elements play a role. In particular, mast cells, eosinophils, T lymphocytes, macrophages, neutrophils, epithelial cells. In susceptible individuals, this inflammation causes recurrent episodes of wheezing, breathlessness, chest tightness, and coughing, particularly at night or in the early morning. There's more. These episodes are usually associated with widespread but variable airflow obstruction that's often reversible, either spontaneously or with treatment. The inflammation also causes an associated increase in the existing bronchial hyperresponsiveness to a variety of stimuli. Reversibility of airflow limitation may be incomplete in some patients with asthma. Okay, so it's defined. There you go. Obviously complicated, and even from the definition, sounds syndromic, not molecularly defined. The burden of asthma is remarkable. Affects everyone, all races, ethnicities. Estimated that 8% of American adults and 10% of American children have asthma. Globally, as many as 300 million with asthma. The whole lifespan, infancy to old age. Before puberty, it's boys more so than girls. After puberty, women more than men. And over the last few decades, it has increased in its prevalence. This is just one slide from the early 90s, and you can just see, less than 1 year old, 1 to 4 years old. In terms of onset of asthma, this is where it starts in most people, not all. And you can see that even greater than 50, some people have incident asthma. And we see these people in our asthma center, interestingly. But this is the typical incident rates. Key features illustrated here. There's the symptoms seem to result from some combination of airway smooth muscle constriction and bronchial inflammation to produce cough, dyspnea, chest tightness, and wheezing. And you can see the picture of the illustration of the asthmatic bronchiole there. So this is a slide of sadness of a young woman who died with status asthmaticus, and Bobby Padera is our lead autopsy pathologist. So this is unfortunate, but this is no longer an illustration. This is what happens in severe asthma. And in the, let's see if this works. Yeah. In the middle here, you can see a mucus plug. And the airway is not a cylindrical tube, but it's constricted. And so it's got those crenellations in it. And all this mucus cell metaplasia with goblet cells have filled with mucus, just ready to fire. Lots of inflammatory cells. And then there's smooth muscle hypertrophy and proliferation that's constricting the whole airway. And this is a lot of inflammation that's going on here. And so as someone who thinks about stop signals, not only go signals, one wonders whether something this severe and horrible, could it be resulting not just from repetitive exposure to pro-inflammatory factors, but is there possibility that this could be a failure of our resolution mechanisms that typically stop inflammation? There is a lot of construction going on here in Madison. I'm really impressed. Okay, so you're all very familiar with what we think of as active pillars of inflammation, defined in ancient times: fever, erythema, edema, pain, and Virchow added loss of function, lessio functio. We now think of resolution as an active process with specific cells and specific mediators, chemical signals, to remove microbes, dead cells, and debris from inflamed tissue, restore vascular integrity and perfusion, regenerate tissue, remit fever, and relieve pain. It's not just a passive removal of pro-inflammatory factors. So early in my career, I was really attracted to Robinson Cotran as the textbook of pathology and Guido Mino's textbook of pathology. And thinking about leukocytes coming out of the bloodstream, entering a tissue, they get bombarded with lots of signals in our bodies. Go faster, go slower, come, go. But eat, don't eat, etc. Go right, go left. I like this description of that. The cells have to integrate these signals and then respond. And on the cover of Science magazine, now more than 2 decades ago, was this picture of neutrophils. And these are not just spheres like globes or like planets. These neutrophils have been exposed to the chemical signal leukotriene B4 in just trace amounts. And notice the intense membrane remodeling that's going on here as cell membranes are loaded with essential fatty acids that get released upon pro-inflammatory signals, and they work as what's called autocoids. So locally, they're rapidly formed from the parent fatty acid, locally acting, even back on the same cell, and then they're rapidly metabolized so that they're like the rheostat, if you will, of local inflammation. Got me very interested in lipid and bioactive lipid mediators. This is from a review that I wrote with Charlie Sirhan and just recently was published on bioactive lipid mediator production during self-limited inflammation. So typically in health, right? We get an inflammatory response, it resolves. And for the purposes of today's talk, I'm going to focus in on the bioactive lipid mediators, not the peptides or the gases that are also part of this whole cascade of signals. So these are dietary essential fatty acids. You can't make them in your body. You have to get them in your diet. Arachidonic acid, an omega-6 fatty acid, and then EPA and DHA, the two main constituents of omega-3 fatty acids in cold water fish or in some algae and plants. And these can all be enzymatically converted to this whole range of bioactive lipid mediators that I'm showing here. And what we found in a self-limited exudate by sampling the exudate at different time points and then doing mass spectrometry and tracking this metabolism is that the products early during the upslope of inflammation are different than the downslope of inflammation. Same parent fatty acid, but the metabolism changes. And so there's something that we just termed the lipid mediator class switching, thinking of antibody class switching. As arachidonic acid early, for example, gets converted to prostaglandins and leukotrienes, but later it gets converted to lipoxins. And we found that some of the signaling mechanisms that convert arachidonic acid metabolism from a go signal to a stop signal, and these are all structurally and functionally distinct bioactive mediators, different functions. So during the initiation and leukocyte trafficking of an inflammatory response, we see a collaboration between the leukocytes and the structural cells of wherever they're entering. And then that evolves in these class switching events to resolution. So this is just some of now a large class of bioactive lipid mediators that are involved in stopping inflammation. Specialized pro-resolving mediators, SPM for short, and they engage specific cognate receptors. And this is an area of active science. We're still identifying some of these receptors. And these, you can see the receptor binding affinity is in the low nanomolar range. These are very potent, but again, they're very locally acting. So not high concentrations in the plasma or in serum, unlike some of the cytokines. And then the receptors are expressed on specific cells, and you can see that for each of the receptors, there's some cells that predominantly express them. And that helps to direct cell type specific actions of these bioactive metabolites. And macrophages are kind of the janitors, if you will, of the lung. And one of the things they do is they eat things, whether it's bacteria or debris or apoptotic dead cells. And you can see that these molecules signaling through these receptors in low nanomolar range actually can activate that resolution function of macrophages, and it helps to define them as a pro-resolving mediator. So from resolvin D1, RVD1 for short, Resolvin D2, Protectin D1, and Merizin 1. These are all part of a DHA metabolome. Again, omega-3 fatty acid metabolized to these molecules. And I'm going to focus in mostly today on the DHA-derived molecules and spend less time on the others. And in addition, these are all lipids with very precise double bond geometry and chirality for the hydroxyl groups. There's also conjugates where peptide from glutathione interacts with the lipid to form a conjugate. You are aware of this in leukotriene D4, that's a cystenyl leukotriene. It turns out there's cystenyl SPMs as well. So I'm going to show you, this is one of the newest areas of science. I'm going to show you a couple stories from that. Okay, so really nice population-based study in children, the COPSAC trial in Denmark, published in the New England Journal in 2016, found a relationship between fish oil-derived fatty acids in pregnancy and wheeze and asthma in offspring. And the bottom line of this study was that supplementation with omega-3 long-chain polyunsaturated fatty acids, so EPA and DHA, in the third trimester of pregnancy reduced the absolute risk of persistent wheeze or asthma and infections, respiratory infections in the lower respiratory tract in offspring by approximately 7 percentage points, which was a third of the risk. So this was very exciting, and it led to Chris Ramsden writing a kind of an associative perspective along with this. And in red there are conditions for inflammation. That's the arachidonate story going to leukotrienes. And in blue are the fish oil-derived resolution of inflammation and bronchoconstriction. These are DHA and EPA-derived SPMs. And so the mechanism of action for this benefit in the offspring was thought to be related to an enhanced formation of these SPMs in the offspring. So today I'm going to focus in on a few vignettes of emerging science around targets for these SPMs in what we think are important cells in the asthmatic airway. Airway smooth muscle, airway epithelium, the leukocytic infiltrates, and regulatory T cells specifically that regulate T cell activation. Okay, so how do we establish the correct diagnosis in asthma? Three main things: a characteristic history, characteristic exam, and some diagnostic testing. Of course, on history, we look for episodic symptoms, characteristic triggers, and characteristic responses to medications. On the exam, we actually still use our stethoscopes, not just a point-of-care ultrasound. We listen to the chest, and we hear wheezes, and then we do diagnostic testing, pulmonary function testing. This is an example of somebody with asthma having an obstructive pattern to their spirometry. North is expiration, south is inhalation. And you can see a peak expiratory flow rate. And then in health, you would connect the dots that are labeled there, but you can see that the airway flow collapses early and has a coving pattern to it, very typical of airflow obstruction. And what we typically see is a reduced FEV1, relatively preserved forced vital capacity, FVC, and a reduced ratio. And that's how pulmonologists and allergists often diagnose obstructive airflow obstruction in their patients. We used, in this set of experiments, precision cut human lung, so we could look at contraction of the smooth muscle. And this, Raja Abdulnoor in the lab led this series of experiments. And on the vertical axis here is leukotriene D4 stimulated airway contraction. And you can see on the horizontal axis in log scale concentrations in nanomolar amounts of leukotriene D4. And so in the dish, we can watch the airways contract. And on the bottom there in the gray, VEH is vehicle. And so you can see, if you connect those black circles, we got to about 50% contraction of the airway. So it's pretty significant contraction. And then these Merizin conjugates that I mentioned from DHA could blunt that LTD4 mediated contraction. MCTR3 in particular could do this. So this is for airway smooth muscle traction. We also do something called bronchial challenge testing for people that were not sure of the diagnosis, and we look for airway hyperreactivity. This is typically done with methacholine when we administer it by nebulizer in increasing concentrations, stopping after each one to check spirometry to see if there's been a change. And the degree of airflow obstruction present after any given concentration is a reflection of the airway reactivity to this cholinergic stimulus. And so if you have hyperreactive airways, you constrict at really low concentrations. You're very sensitive to it. And so this is a typical bronchial challenge test. You can see forced expiratory volume in 1 second on the vertical axis. And then in each of those red diamonds is a concentration of methacholine starting from 0, 100% of baseline, and then going up. And then this person got to a 20% decline at 6.2 milligrams per ml of methacholine. And so the provocative concentration for a 20% decline is 6.2 milligrams per ml. That is a hyperreactive. Anything less than 8 milligrams per ml is defined as hyperreactivity. This is another person who went through the same protocol, who had a little bit of a dip, but never got to 20% drop. This is a non-reactive test, so you can't really define a PC20. So this is how we do it in humans. We have a whole series of experiments that we do in mice, and we can't get mice to use a spirometer. So we have to do a different way of assessing this, airway hyperresponsiveness. We put them on a ventilator, and we actually measure lung resistance of the respiratory system. So this is mice that get exposed to chicken ovalbumin. They've never seen it before. They get sensitized to it, and then we aerosolize them daily for a few days so that we have an allergic airway response to the allergen. And then after the animals are fully inflamed to the max, that's when we give them either a vehicle or these MCTRs, these SPMs, and look and see what happens. This is our parameters. Naive animals in the white, you can see they don't really respond much to methacholine, but this is resistance to the respiratory system. But when they've been sensitized and challenged, they're quite responsive to methacholine. And then here's the response when they're exposed to the MCTRs. You can see that this is driving from the highly sensitized all the way down towards naive. And MCTR3, again, is the most potent at doing this. This is the molecular structures. And so this is promoting, remember, we didn't start these until after the animal was fully inflamed. So we're promoting resolution of airway hyperresponsiveness. And I'm not showing you the data, it's in the publication about inflammation and leakage permeability, the edema that we see. This set of experiments was picked up by the New England Journal for their clinical implications of basic research, balancing the effect of leukotrienes in asthma. The biosynthetic pathway is shown there. DHA, the parent fatty acid, and this is a 12-lipoxygenase initiated pathway. And this is an illustration of the experiments I just showed you. And so we have a drug that we will turn to at times in our armamentarium to treat human asthmatics, montelukast. It's a cis-LT1 receptor antagonist. It's blocking cystinyl leukotrienes. In most people, it's not particularly effective. These sets of experiments uncovered the fact that actually endogenously you have already the drug. You have, if you're converting DHA to these molecules, they're blocking LTD4. So the addition of a cis-LT1 receptor antagonist doesn't do a whole lot. If you're deficient in DHA, it probably is much more effective. So this helped to understand the science in our lungs of what was going on. That was a 12-lipoxygenase derived set of molecules. This, I'm not going to go through this in detail for this talk, but you'll see DHA on the far left, and 15-lipoxygenase is now the initiating enzyme. And you get protectin D1 and the PCTRs, protectin conjugates. And I want to tell you a story about this set of molecules. So protectin D1 you can detect in just human exhale breath. And in model systems, it's a potent regulator of allergic lung inflammation. In influenza viral infection, protectin D1 can actually inhibit replication of the virus. And we know that these PCTRs are in lung. We've detected them in human lung. So I wanted to tell a story about respiratory syncytial virus. It seems like it's the season, so thought I'd bring this to you today. And for this, we're using a clinical strain of RSV. It's called line 19 from Nick Lukács at the University of Michigan. And we're giving it to them, we're inoculating the mice. And what happens is we see the whole arc of inflammation and resolution around the airway, typically. And this is what the histology looks like. The airways get very inflamed, and especially around the epithelial cells. And you can see by day 12, they're getting closer to what a normal airway would look like. Placable virus peaks at day 6 after inoculation and is coming back down by day 12. And then cytokines are being released in response to the viral infection. I'm just focusing on IL-13 here, one of the T2 cytokines. It starts to go up at day 3, peaks at day 6, similar to the virus. And IL-13 is being produced by 2 different cell types, an innate lymphoid cell, type 2, which is shown on the left, these lineage negative cells, and then on the right is a helper T cell. And you can see the innate cells already present in your lung kind of ramp up a little bit faster on day 3 than the adaptive immune response, where your T helper cells are coming up a little bit later, but both peak around day 6 in terms of IL-13 expression. Okay, so in that model, we inoculated the animals, and then Kate Walker, who's pictured here, led this story about, which ultimately ended up being about airway epithelial cells. We waited until day 3 after inoculation, and then we either gave PCTR1, the protectin conjugate, or protectin D1, or vehicle. We gave it in the nose. And then at day 6, we looked for the outcomes that I was just mentioning, viral RNA, inflammation, et cetera. And first weeks, what we saw by PCR, these 2 important genes in the RSV genome were actually less present by PCR with PCTR1 and PD1. So there's less presence of the virus. At the tissue histology level, RSV and vehicle is shown, and that's the second sets of images, 10x and 40x. And then with PCTR1 and PD1 below, you can see that there's less inflammation. It's pretty evident. When you actually quantitate eosinophils and neutrophils, there's less of them in the airways. Natural killer cells and even activated NKG2D positive NK cells, there's less of them, in particular with PCTR1. One of the antiviral peptides for host defense is cathelicidin. This is RNA for that. And that's actually with the vehicle, you can see it's reduced with RSV infection, but ramped up with PCTR1 and to some extent with PD1. And then we looked at interferons. We looked at T helper cells that were producing interferon gamma, and they get markedly increased with the RSV infection, and you can see they're reduced, again, especially with PCTR1. And interestingly, we looked at interferon gamma levels and found that those were decreased with infection, but actually increased with PCTR1. At least there was a trend there. So we cultured human airway epithelial cells, infected them with RSV. And what we found is with vehicle, we don't see much of an interferon gamma signal from the airway epithelial cells, but with PCTR1 and PD1, we saw a marked induction of interferon gamma. So this was pretty cool, direct actions on the airway epithelial cells. Those are the first molecules, endogenous molecules, that are known to actually ramp up interferon gamma. And so as the inflammation is decreasing from the RSV and the amount of RSV is decreasing in the presence of these molecules, we're seeing interferon gamma ramp up. And interferon gamma is just, it's our, in this sense, one of our first lines of defense against viral infection. It's produced largely by the airway epithelium, and it doesn't activate a whole inflammatory cascade. Unlike the type 1 interferons that we became very familiar with during COVID, where not only do you get the airway epithelial expression, but you get a ramping up of intense inflammation behind it. So the more gamma we have, the less reliant we become on our second line of defense, the type 1 interferons. This became quite relevant in COVID. And this group of investigators published in Lancet Respiratory Medicine their phase 2 trial And here they're looking at the proportion of patients negative for SARS-CoV-2 RNA in the presence of viral load, that relationship. And they used PEG interferon lambda as an injectable versus placebo. And what you could see is that the probability of clearance of SARS-CoV-2 by day 7 was, especially with higher viral loads, was protected by PEG interferon. This led to a phase 3 trial published in the New England Journal of Medicine. In orange here is the PEG interferon lambda, in blue is the placebo. And they looked at hospitalizations and ED visits. They looked at whether you were vaccinated or not, and they looked at COVID-related deaths and severe illness, all of which were protected with PEG interferon lambda. So the body, responding to a viral infection by ramping up interferon lambda is a very important protective action. So this is clinical translation of that observation. I just want to show a brief slide here, just for sake of time, on regulatory T cells. Nandini Krishnamurthy in the lab looked at the same RSV infection that I was just telling you about, that model. But here she's looking at Merizin-1, and Merizin-1 did not induce interferon lambda. This is another DHA metabolite. It just worked differently. What it did was it activated regulatory T cells to a more suppressive function. It led to a proliferation of the regulatory T cells, and it corrected their actions from pro-inflammatory to pro-resolving. And so you can see with MAR1, the airway looks much less irritated and inflamed than with vehicle. And when we looked at IL-13 from either innate lymphoid cells or type 2 helper cells, the Merizin-1 exposure decreased IL-13. And there's more in this publication. But this was exciting because, again, it's an endogenous molecule identifying a regulatory mechanism for cells to control both innate and adaptive inflammation to a respiratory virus. Those type 2 cells are very important in many asthmatics. This is early work in the New England Journal from Dwight Robinson and colleagues, and they did BALs on asthmatics and did autoradiographs for antisense riboprobes for IL-5, IL-4, and GM-CSF, type 2 cytokines. And they also then looked with beads that were tagged with CD2 and found that these were being produced by T cells. So these were adaptive T cells in the asthmatic airways in asthma. And over a period now of many years and a lot of work, this is the type of paradigm that has been developed for activating those type 2 helper cells. You get exposed to antigen. Antigen-presenting cells in the airway through MHC class II can pick up, can recognize that antigen, and then educate naive T cells, here Th0s, in particular towards a type 2 Th2 phenotype. And those cells then produce IL-4, IL-13, which you can see have many of the asthmatic features to them, IgE synthesis, mucin secretion, et cetera. And then also IL-5, which leads to eosinophil activation and eosinophilic airway inflammation. So many asthmatics have type 2 inflammation, and that's in the pink here, in the red. Some of it is a lot of type 2, some of it's just more modest type 2. So I'm showing a gradient. This is a slide courtesy of John Fahey I really like. But there are also asthmatics who really don't have much in the way of type 2 mechanisms going on. They're type 2 low or no type 2. So this is, I think, a really helpful framing of heterogeneity in clinical asthma. And it argues really against a single disease mechanism, that it's really more syndromic, that asthma is more of a clinical, physiologically defined syndrome. And so this is a review article that was published in 2022 in the New England Journal, and it looks at what we know now, what's our current state of information about airway inflammation in severe asthma, and targets of now this proliferation of biologics that are being used to treat asthma. And small print, lots of cells, lots of molecules. Don't expect you to be able to read that Across the top in red there is type 2 high asthma. And on the right, in the kind of greenish color, is type 2 low asthma. The width of the bar for type 2 high asthma is much bigger. So, and that reflects, I think, the population of asthmatics that we care for. And all of those white boxes are drugs that have been developed against specific molecules in this cascade of inflammation. If I get rid of the type 2 high asthma, there's no white boxes. We have no treatments for people that are in this camp. And many of our asthmatics don't respond to those biologics. Or if we stop those biologics, they're not cured. The disease hasn't been modified. The type 2 inflammation just comes right back. So those are really potent controllers, and some people benefit tremendously from them. But there's a whole area of severe asthma or difficult to control asthma that they don't cover. So it's just, for those of you who might be interested in this space, you've got role models and mentors right here in Wisconsin, and there's lots of work still to do. We see a lot of neutrophils and other kinds of helper T cells, Th1, Th17, mast cells, that are part of this low signal. The syndromic nature of asthma comes out in the study of genetics of asthma as well. The genetics of asthma are notable for being associated with SNPs, single nucleotide polymorphisms, in several genes. So there's already kind of a clue that it's more syndromic. And there are only very few that are associated with all ages, and those seem to be related to epithelial damage and activation of inflammation. And the loci that are strongly associated with IgE, atopy, are generally not significantly associated with asthma. So this is from the New England Journal, from a genome-wide association study. And what you can see here are several loci that have genome-wide significance. So that was pretty cool. And this is the table and that accompanies this article. And in the top there are loci that were linked to serum IgE levels. And in the bottom are loci linked to asthma. And there are very few that are the same. IL-13 is the same, but very few others. So this already tells you that this is quite syndromic. And we all know that there's important environmental influences in asthma as well. Just people are allergic to things in their environment, et cetera. So there's a gene by environment interaction that's very important in the clinical expression of asthma. Well, the genetics that I was just mentioning came home for us in the story about resolution when this group of investigators at the UK Biobank looked across their enormous data set to try to prioritize candidate causal genes for asthma and susceptibility loci. And this was in one of the Nature journals. And one of the genes that they found that of interest was this GPR18, which has been defined now as a receptor for resolvin D2, one of the DHA metabolites. So we wanted to study this in more detail. So DHA enzymatically converted by a 15-lipoxygenase and a sequential action 5-lipoxygenase forms a series of D-series resolvins, and I've circled resolvin D2. And this has been, prior to this work that I'm going to share with you, us and others have looked at this in the role of resolution of infectious inflammation, controlling NF-kappa B and linked to severity in sepsis, and both in vivo in animal models, but also in vivo in humans. And then this bottom article is about the identification of DRV2, the receptor for resolvin D2. And it was looked at in infectious inflammation. So Hong-Yong Pei in the lab led this series of experiments where we're using house dust mite extract. And so we're sensitizing the animals to house dust mite, and then we're challenging them again in the airway. And then once they're fully inflamed, we would give them either vehicle or resolvin D2, and then we could track what's happening over the ensuing several days. I think it's a good point in the talk. We're deep into the talk now, 40 minutes in. And to take a moment, I think we have a minute or two to just reflect for a minute on the social context of asthma. We're talking about house dust mite. It's an indoor allergen. This schematic is Boston. These are neighborhoods in Boston. And in the dark brown are neighborhoods where the prevalence of asthma is higher. So this is tracking asthma ED visits for kids aged 5 to 17. And so the visit rates are much higher in the dark neighborhoods. So you can see there's an intermediate brown, and then there's areas that have lighter brown. So why is there a difference in the neighborhood prevalence of uncontrolled asthma? Anybody have any thoughts about that? I present data from the New England Journal and from these very, detailed animal models. It should work, it should work, it should work. But we see this in real life. This is real life asthma, right? This is the home of the Inner City Asthma Consortium. What's going on? Why are we seeing this? We really have to be careful about these types of talks and then implementing them in our care of our patients. Because in addition to kind of scientific points of advance, there's financial and non-financial barriers to care. And indoor allergens like house dust mite and cockroach and mouse may be much more active in certain neighborhoods where people have less control over their indoor environment, for example. So especially in cold winter wonderlands like here, like Boston, we need to be careful about this. I thought it was important to put this in social context. Okay, so here's the impressive data. We're going to focus in on leukocytic infiltrates. And in the red are the animals that got resolvin D2. In the black are vehicle. And you can see whether you look at day 17, 19, 21, there's an advantage with resolvin D2, in particular regulating eosinophil and lymphocytes, especially T cells. When we think about resolution, we've tried to develop metrics to quantitate whether we're having a positive or a negative influence on the endogenous resolving mechanisms. And so we track over time here, and here we're looking at eosinophils in the BAL of these, in the whole lung lavage of these animals. And in vehicle, you can see from day 15, which is right when we start giving resolvin D2, the eosinophilic inflammation goes up, actually, before it comes down, whereas those animals receiving resolvin D2, it comes down right away. And we think about a resolution interval as the time it takes to go from the max to the half max, or from the initiating point to at least halfway down from there. And so in this set of experiments, it takes 7 days to really see that eosinophil in the BAL counts go down by half from the vehicle, whereas we're seeing it very rapidly within 1 day with resolvin D2 on board. This is really evident in the histology of the lungs of these animals. Again, the animals were fully inflamed, and then we give either vehicle or resolvin D2, and you can see the intense inflammation and changes to the airway compared to the saline control in the model. And you can see it's much less prevalent with resolvin D2. If you look at mucus by a PAS stain, you can see there's mucus cell metaplasia in day 17 in the model, much less prevalent with resolvin D2 on board. So you can see there's been a big acceleration of resolution with therapeutic administration of resolvin D2. This is the airway hyperresponsiveness, and you can see the naive in the dashed line, and at the top there, the vehicle, and there's been a modulation down by resolvin D2 at day 17, and if you go further out to day 21, it's now at baseline. It's back down to naive with resolvin D2. You can see it's still elevated with the vehicle. We can quantitate this by looking at what's called the effective dose to lead to a doubling, a 200% change in the respiratory resistance. And in red there, it takes more methacholine to double the airway resistance than in the vehicle. And then you can see by day 21 in red there, it's the same basically as naive. So there's been a return, if you will, to normal airway hyperresponsiveness. So an acceleration of that resolution. Type 2 cytokines, IL-4, IL-5, IL-13 are all decreased by resolvin D2, as is total IgE in response to the house dust mite. We found the receptor that is of interest, this DRV2 receptor, and there's a competitive antagonist that's been identified called O1918 for the receptor. And what we found is that all the advantages with resolvin D2, this is just showing BAL, total cells, eosinophils, neutrophils in blue here with the presence of the receptor antagonists are blocked. You lose all the advantages. So this is a resolvin D2 DRV2 receptor coupling that gets disrupted by this receptor antagonist. Peripheral blood from human subjects, healthy or asthmatic subjects, are shown here, and I'm just emphasizing the kind of asthma relevant cell types, eosinophils, CD4 T cells, CD8 T cells, B cells. And you can see that there's an expansion of pretty much all of these cells in asthmatic blood by cell number. When we look at the amount of expression of DRV2 in these different cell types, in particular in the purple color, we can see that eosinophils in asthmatics have a marked increase in the expression of DRV2 relative to the other cell types in health. Eosinophils are really cool. I know that they're beloved here in Wisconsin, so I had to say a word about this. So Paul Ehrlich is really in this paper about the specific granulations of the blood. Also, he gave this wonderful presentation in Berlin. I wish I would have been there, 1879, where he mentions eosinophil for the first time and links it to parasitic infection and asthma, and what was known as asthma. And it's viewed, because of its staining techniques, the staining characteristics, as a homogeneous cell population with a role in inflammation. So Tazy Brueggemann in the lab wanted to look at this in a little more detail, especially in context of the resolvin D2 studies that I just mentioned. So we looked at spatiotemporal regulation of the eosinophils here. It's the house dust mite model. We followed out a little bit longer. And instead of just looking at the BAL, like in the study I showed, we look at total lung eosinophils now. And we see a different kinetic. If you look at the BAL, it's in red. That's what I showed you before, day 15 going up at day 17 and coming down. But if you look at total lung, there's like a different set of kinetics. It looks like it's regulated in a different manner. So we use flow cytometry to try to understand some differences of what might be going on in the lung versus BAL. And I'm not going to go into the details of the immunology here, but we kind of zoom in by flow cytometry on eosinophils. And this marker, CD101, not sure of a function, but it's a marker for eosinophils. It turned out that if you look at lung eosinophils, there's a CD101 high subset and a CD101 low subset. And if I just walk you through this, so we're looking at lung and BAL. This is day 0 in the model. CD101 low are prominent in the lung. There's not a lot of them, but what's there is CD101 low. There's no CD101 high, and in the BAL, there's no eosinophils. In that model, as we're getting more and more inflamed, you can see that there's now CD101 high on the right in both of these, in the lung and in the BAL. There seems to be more of them relatively in the BAL than the lung. And when we get fully inflamed, now what we see is entirely a CD101 high subset in the lung, in the BAL. And now you see this really cool bifurcation in the lung, CD101 high and CD101 low. If we look at the morphology of these cells and their transcription by the inflammation panel on RNA kind of transcriptome look, the morphology looks different for the low and the high cells, and the transcription is quite different. This is at day 0 and day 17 only in the CD101 low cells. And you can see there's very significant differences. This is on a nanostring panel, including 15-lipoxygenase, 5-lipoxygenase, COX-2, some of the important biosynthetic enzymes we've been talking about in grand rounds today. Those change, they go up at day 17. And then if we look at the CD101 low versus the CD101 high, we also see very important differences in the transcription of these two different types of or subsets of eosinophils, including in those same lipid modifying genes. So this was, we thought, pretty cool. So we looked to see what happens with resolvin D2 in this setting. And what you can see is that it affects both subsets because the total lung eosinophils go down. I'm not going to show you all the data, but it's really prominently affecting the CD101 high eosinophils. We looked at the regulation of eosinophil function by resolving D2 in this set of experiments. And CD11B is an activation marker for eosinophils. It's higher in the CD101 high cells than the low. And in the blue there, you can see that there's some regulation of this activation marker by resolving D2. This is all at day 17 in the model. This is one of the receptors for prostaglandin D2, a mast cell product in the human airway. And the CD101 high eosinophils express much more of this receptor than the CD101 lows, but we don't see any real regulation of the receptor expression by resolving D2. We looked at function, and that's not erythropoietin, that is eosinophil peroxidase release, so degranulation of the eosinophils. We really didn't see a response even to PGD2 in the CD101 low cells. However, when we looked at the CD101 high cells, prostaglandin D2 elaborates and releases eosinophil peroxidase, and this is partially blunted by resolvin D2. So it's regulating function of the eosinophils. In the severe asthma research program, we've been tracking plasma levels of resolvin D2 in the human severe asthmatics, and they're less prevalent. There's less lower amounts than in the healthy controls. And so I come back to this model, the structural model of the airway. I've shared with you several different scientific vignettes. The MCTRs and airway smooth muscle contraction, the PCTR1 and PD1 on the interferon lambda story, targeting airway epithelium, resolvin D2 and the leukocytic infiltrates, eosinophils in the asthma models, and Berezin 1 and the regulatory T cells I referred to. So these are how we're starting to think about how a parent fatty acid, DHA, could get metabolized to protective, bioactive lipid mediators locally acting in the airway to help in health quiet down inflammation in the airway in the setting of asthma. So I want to summarize the talk, and then we'll open it up for questions if there are any. Here we've presented evidence that for allergen or viral-driven type 2 airway inflammation, SPMs, specialized pro-resolving mediators, accelerated the resolution of lung inflammatory cell counts, type 2 cytokines, IgE, mucus cell metaplasia, and airway hyperresponsiveness. The actions were receptor dependent, and receptor expression was modulated on the leukocytes. I showed you the human data, not the mouse data today. After HDM, the house dust mite, the lung mouse eosinophils were spatiotemporally regulated with an activation phenotype distinguished by CD101 expression. And the CD101 low and high EOs were morphologically, transcriptionally, and functionally distinct. Resolvin D2 regulated eosinophil numbers, transcriptome activation, and function. So in conclusion, unraveling this endogenous system of resolution has revealed that resolvin D2 is a potent SPM agonist for mouse type 2 cytokine evoked allergic inflammation, regulating IL-4 linked to the regulation we saw in IgE levels, IL-5 regulation of eosinophil, IL-13 regulating the mucus cell metaplasia, the airway hyperresponsiveness, and that resolvin D2 seemed to regulate eosinophil-specific phenotype and function. The actions were DRV2 dependent, and expression in the leukocytes suggests that this receptor could serve as a regulatory target for asthma. This is from Scientific American in an article that Charlie Serhan wrote about this class of molecules serving as immunoresolvent agonists for catabasis. Catabasis is a military term returning the battlefield back to kind of a non-wartime state. So thinking about the lung or the airways as a inflammatory battlefield and kind of returning that to a quiescent homeostatic state. We're comfortable thinking about drugs that we use clinically, like in anti-TNF molecules or even NSAIDs, as inhibitors of distinct inflammatory pathways. There's other biologics, or anakinra, even cyclosporin and IL-12. These molecules are serving as agonists, not antagonists, at specific receptors, and they block very potently activation of granulocytes, neutrophils, eosinophils, and in a non-phlogistic way, they activate macrophages and monocytes. I haven't shown you that data so much today, but I did mention the macrophage phagocytosis function at the very beginning. So I get the privilege of working with a really wonderful team, many of whom are shown here. I've tried to highlight people's contributions along the way, and many of them are in bold here. Charlie Sirhan is actually the person who discovered all of these SPMs and their formation, their sites of action. They're a wonderful collaborator. Elliot Israel and his team in the Asthma Research Center at the Brigham. I'm a member of that, and he's been a wonderful collaborator over the years. And our severe asthma research program, Bill Bussey started, and Lauren and I get the pleasure of working in, along with Nazar, his amazing colleagues in this type of work, and our funding from the NHLBI. And I'll just end with the session evaluation. Thank you very much for your attention.
Great. Thank you for a wonderful talk. Really love kind of how you used what we see clinically in our patients to drive the research and going from bedside to bench and then hopefully bench back to bedside and beyond. I'm going to ask my question. So what struck me is the resolvins that you're talking about are all endogenously produced molecules from lipids that require exogenous, we need to get from the outside. We don't make them ourselves. So my question is, and I also was struck by your lipid class switching. So the question is, what is failing in severe asthma? If we are able to produce these molecules that can resolve inflammation, is it that this class switching doesn't occur enough? Is it overwhelmed? Is it lack of initial substrate? So
Wonderful, wonderful question. And we don't have complete answers, but there was a really interesting study done for cystic fibrosis where Mike LaPosada and colleagues biopsied the mucosa of the cystic fibrosis airway and looked at omega-3 fatty acid content, arachidonic acid content in the mucosa of this inflamed airway. And what that group of investigators found, it's published in the New England Journal, is that there were lower amounts of DHA in the airway in cystic fibrosis. And I'm guessing the editors made them do disease controls. So in that paper, they looked at asthmatics. And it turns out that DHA content in the asthmatic airway is abnormally low. And so the COPSAC trial, which I showed you for supplementing mothers and then looking at offspring, the most prominent benefit was in the group that had the lowest quintile of DHA content in their, they measured DHA levels in the blood. And that's the group of mothers that benefited the most for their offspring with the supplementation. That lowest quintile in Denmark is higher than the highest quintile in the United States in a Western diet. So I think that in general, we don't get enough of this in our diets. In inflammation, it is consumed in the airway, and it's not clear yet how robust one has to be to feed the lung through the gut. Those are details that have not been worked out well. There's been some work about DHA transporters like in the retina, because most of the DHA in our body is actually in neural tissues. That's the highest, the brain has the highest content, but the retina is really high. There's been a few clues about this, but not in the lung yet. We need to figure that out. But I think it's largely related to parent availability of substrate. Yeah.
So thank you, Bruce. And just going to summarize some of the online comments as well as I'm feeling really good that I had salmon last night for dinner. So, should we all be running out to get fish oil or, are there, and you kind of alluded to this with the gut system, are there any airway microbiome contributors that might make agonist for the resolvin D2 receptors?
Yeah, so the microbiome is not entirely uncharted territory, but there's been some intriguing data related to the microbiome and production of molecules that can engage these pathways. So more to come there. We're not actively doing that ourselves, but there are others who are actively looking at that. And it's really an interesting but still evolving area. We don't have definitive information. I'm not going to advise what you should do with your diet, but do I take omega-3 fatty acids? Yes. But you have to be careful about this. It's unregulated, right? And so like a banana that turns from yellow to brown pretty quickly, those double bonds in those parent fatty acids can get non-enzymatically oxidized, and then you disrupt your ability to form these molecules. So what I would advise is keep it in that dark brown bottle. Don't buy a lot of it. Buy some and then use it up and get some more. And don't let it sit on the shelf very long. And there are some that are better than others, but we've actually looked by mass spec and kind of contaminants and stuff like that. So it is worth spending a little bit more to get a high quality product. No brand names.
Great. Well, thank you so much. I really appreciate it. And thanks for the audience for your attention. And stay safe today. I think it's supposed to snow a bit later, so drive safely. And thank you again. Love to have some more interest in asthma research. We've got amazing researchers here. So just making a plug.
Happy Valentine's Day, everyone.
And yes, happy Valentine's.